Hi everyone, today we'll be talking about recombinant DNA. This project is done by me, Sarah Mohsen. Before we start, we'll have to review some vocabulary words. The first one is plasmids. Plasmids are small circular double-stranded DNA molecules that occur naturally in bacteria and yeast cells. They can be used as vectors or carriers because they can be cut with restriction enzymes. Let's have a closer look at the plasmids. Plasmids have a target gene, which is the gene we want to make copies of, and an antibiotic resistance gene. This we'll learn about it later in the video. The next word is restriction enzyme. Those are enzymes that recognize and bind to surface DNA sequences and cleave or cut the DNA within that sequence. They're also called an endonuclease. Notice that it ends in ase, which is a hint that it's an enzyme. Fun fact, scientists have identified and isolated hundreds of restriction enzymes. Restriction enzymes act like scissors. The last word is DNA ligase. They join two different fragments chemically. It joins DNA fragment that has sticky ends. They are used for repair and replication of DNA. Now let's start explaining what is recombinant DNA. Recombinant DNA is a technique in which a fragment of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it. So basically, recombinant DNA is a method where scientists can add any gene they like to a gene that did not have the gene they're adding. Now we'll see the steps of making a recombinant DNA. The first step is we're going to take our gene of interest from its DNA and cut it with restriction enzymes. We use the same restriction enzyme to cut open the plasmid. Here's a question: why do we use the same restriction enzyme? We use the same restriction enzyme to create complementary sticky ends. If different restriction enzymes were used to cut the plasmid and the gene of interest, they're not going to be complementary and they're not going to match. After inserting the gene of interest into the plasmid, the DNA ligase can do the job of sticking them together. It shouldn't be hard sticking them together since the same restriction enzyme was used to create the complementary sticky ends. Finally, the plasmids are mixed with a solution containing E. coli. The bacteria now can take in the plasmids and replicate. But not all bacteria will take in the plasmid, so we can't find which bacteria has the plasmid and which does not. That's why the solution is displayed in a plate containing nutrients and antibiotics. The nutrients will help the bacteria containing plasmid to grow and replicate. However, the bacteria that do not contain plasmid will not survive because of the antibiotic. That's when the antibiotic resistance gene takes place. The plasmid contains a gene that resists antibiotic and can grow even with the existence of antibiotic. Let's recap over the steps. Firstly, a DNA fragment containing the gene of interest is obtained from a donor cell. Then, a suitable plasmid is obtained from a bacterium. Thirdly, the DNA fragment containing the gene of interest is cut using restriction enzyme. This enzyme acts like scissors. On the other hand, the same restriction enzyme is used to cut open the plasmid. Lastly, the DNA fragment containing the gene of interest is inserted in the open plasmid with the help of DNA ligase. It catalyzes the joining of the DNA fragment and the plasmid. A recombinant plasmid is finally formed. This recombinant plasmid can be introduced into a host cell. But what's the use of all of this? Why is recombinant DNA so important? Now we're going to learn about the uses and aims of recombinant DNA. Number 1, it can be used to produce drugs or vaccine like insulin. Before recombinant DNA was discovered, insulin was extracted from animal pancreas tissue to treat diabetics. Now insulin is produced in bacteria using recombinant DNA. First, find a gene that codes for insulin protein, and bacteria produces insulin for us so we don't have to take pancreas tissues from animals. This is a cheaper way of obtaining insulin. Number 2, recombinant DNA can be used to produce things such as HGH, which is a human growth hormone, or blood clotting factors, or hepatitis B vaccine. Finally, recombinant DNA can be used to reduce vitamin deficiency. For example, golden rice. One big difference between golden rice and normal rice is that golden rice has more vitamin A. In parts of the world where there may be nutritional deficiency, the rice is the majority of what people eat. So some people have vitamin A deficiency. Golden rice is there to supplement that to reduce vitamin A deficiency.